Hi, it's Tamlin Oliver, Managing Editor of BioCompare. Welcome to today's webinar on simplifying ChIP from primary cells and clinical samples for epigenetic applications. This webinar will spotlight the research of Tobias Waltermann, a PhD candidate at Bayer. He'll be introduced by Roman Rodriguez, Director of Marketing at Covaris, sponsor of today's webinar. Before I pass the mic to Roman, though, I want to review a few webinar logistics. We'll hold a live Q&A session at the end of the presentations. You can submit a question at any time using the Q&A box on your screen. Also, your webinar console is fully customizable, and we encourage you to move, maximize, or minimize any of the tools on your screen to optimize your viewing experience. There are also a number of resources to the right of your screen available for your review and download. If you experience technical difficulties at any time during the broadcast, there's a help button on the bottom toolbar of the screen. Ping us and we'll do our best to assist you. Finally, today's webinar is being recorded and you will receive an email about 24 hours after the event today advising you the webinar is available on demand so you can view it again or share it with colleagues. All right, that's it. Let's get this webinar started. Roman, take it away.
Hello everyone and welcome to our epigenetic focused webinar developed in collaboration with Bayer AG. Today's webinar is brought to you by BioCompare and sponsored by Covaris. I am Roman Rodriguez, Marketing Director at Covaris, and I will be moderating the presentation. After a quick introduction to Covaris technology, I will hand it over to today's presenter, Tobias from Bayer AG, who will be discussing the standardization of a ChIP workflow from challenging samples like primary cells. Our webinar will be interactive, so please submit your questions or comments as we will be handling a Q&A session after the presentation. Dr. Angela Gardings, Field Application Specialist at Covaris, will be then joining us for addressing the technical aspects. Let's start with a brief introduction of Covaris technology. AFA, that stands for Adaptive Focused Acoustic, is a technology that Covaris developed and implemented 20 years ago to applications like DNA sharing, and now implemented in many different new areas. There is a range of systems capable of treating one sample at a time to high throughput, fully automated, thanks to recent developments in the systems, the consumables, and reagents. Our mission at Covaris is to help accelerating discoveries by developing innovative pre-analytical sample prep solutions. We want with our technology to simplify and standardize clinical sample prep, increase quality result while achieving superior throughput and automation. The AFA technology is based on ultrasonic acoustic energy, generating a cavitation-based process. To simply describe the cavitation process, the acoustic energy traveling through the liquid medium will create pressure waves, like you can see on the diagram, causing the dissolved gases to come out of solution as microscopic air bubbles. When these bubbles implode, like you can see on the diagram, the surrounded fluid rushes back into the field void created, and that results in extreme shearing forces. Our technology is highly tunable, and we can have a tight control of the bubble's density and the speed at which they implode. Another key aspect is that the medical-grade transducers generate ultra-high frequency energy, resulting in short wavelengths, and thus a more focused tone around 3 millimeters. So because the energy is not dissipated but focused on the sample, we need less energy, and we can proceed in another thermal environments, as you can see on the diagram on the bottom right. The temperature control and regulation is key, as it could be detrimental to your biomolecules. Our equipment can be located at the bench and do not require any hearing protection as they generate almost no noise. Sample preparation involves many various steps, sometimes developed a long time ago and never fully optimized. Biomolecules could be sometimes damaged during the lysis or extraction phase, and it's been documented that detection sensitivity and quality depends on the sample preparation's optimizations. This control of energy with AFAs allows us to apply the technology efficiently to many applications in pre-analytical sample prep, like cell lysis, biomolecule extraction, and sample processing like DNA or chromatin sharing, but also to standardize the workflows. We would be happy to discuss your application, so feel free to contact us. Now, it is my pleasure to introduce Tobias Waltermann, who is currently working on his PhD thesis at Bayer AG in the Department of Mechanistic Toxicology. His PhD project focuses on epigenomic profiling of human pluripotent stem cells, differentiating toward hepatocyte-like cells through investigation of certain histone modifications during the differentiation process. One of the challenges within this project was to develop a robust ChIP assay capable of tracking different histone modifications in a variety of different cell types, development stages, and primary human hepatocytes. Now it is my pleasure to introduce and welcome Tobias Waltermann.
Yeah, thank you, Roman, for the kind introduction. And I will welcome you to today's webinar about standardizing ChIP from primary cells and clinical samples for high-throughput epigenetic applications. So I will start by showing you the agenda for today. I will first introduce you to one of the main scientific questions of my PhD project and the model systems I had to establish to carry out the experiments. And I will then show you the challenging steps occurring during sample preparation and how I tried to solve these issues. I will then show you some results obtained with my optimized ChIP workflow, and at the end I will summarize the talk. So I will start with a so-called PCA to introduce you to one of the main questions of my project. PCAs are used to visualize sample diversity in a 3D space. And here you can see gene expression profiles of different human liver models. These sample groups are divided in two main groups, which are human in vivo liver samples plotted in green and in vitro generated human liver cells plotted in blue. These human in vitro generated liver cells are derived by differentiating human stem cells towards an hepatocyte phage. And we wanted to compare these cells to their in vivo counterparts to see how great they to just see to how an extent they differ from the in vivo samples. And what you can see here is that the in vitro generated cells differ significantly from their in vivo counterparts and more resemble a fetal instead of an adult phenotype because they are more closer to the human fetal sample than the human adult samples or the isolated human hepatocytes, which is in line with the literature. And what we also see is that these cells are generated with 3 different differentiation protocols, and the protocol 3 here is more closer to the human in vivo samples, which tell us that you can optimize the differentiation protocols and that these cells result in a gene expression profile that is more closer to the in vivo samples. So this observation led us to the decision to optimize the differentiation protocols available today to get more cells that are more mature, because these cells that you can generate with current protocols are not sufficient for us because we wanted to do toxicology screenings or disease modeling, and these cells are not suitable for these assays because of the common drawback of these fetal phenotypes. So our hypothesis was that this maturation blockade is at least partly caused by an aberrant epigenome of these in vitro differentiated cells. So one of the main goals of my project is to characterize the differentiation on an epigenetic level and to identify potential targets for improving the differentiation protocols. So this was the initial idea of my project, and I will show you on the next slide the liver development in vivo to just introduce you briefly into liver development and to explain you how we try to mimic this process in vitro. So in vivo liver development starts with the gastrulation. Cells at this stage of development are pluripotent, which means that they can give rise to every cell type of the human body. And one of the earliest developmental processes are the differentiation of these pluripotent cells into the three germ layers, which are the mesoderm, the endoderm, and the ectoderm. At later stages, these endodermal cells give rise to the fore, mid, and hindgut. And we are interested in the foregut cells because they give rise to several organs, including the liver, as well as the biliary tree and the lung, and thyroid, and stomach. So how do we try to mimic this process in vitro? So what we need are pluripotent cells, and embryonic stem cells are a common tool to start from, but they are ethically questionable. So we try to use human-induced pluripotent stem cells. This was a great innovation introduced by Yamanaka in 2006, and he converted somatic cells via transfection of certain factors, and this ectopic expression of these transfected genes results in a transformation of the somatic cell to a stem cell that is pluripotent equally to the embryonic stem cells. So we are starting from these induced pluripotent stem cells and initiate the differentiation towards the hepatocyte phase. What you can see here in these images are cells differentiating towards this hepatocyte phase, which is divided into 4 stages. The first stage is the human-induced pluripotent stem cell stage, so the gastrulation. The second step is the foregut, which is located here. Which is located here. The third stage is the hepatoblast stage, which is located here. So these hepatoblast cells are can give rise to hepatocytes as well as cholangiocytes. So we have to drive these hepatoblast cells towards the hepatocyte phase and to exclude the cholangiocyte phase. And when doing that, you receive hepatocyte-like cells showing the typical polygonal shape of hepatocytes. So the second system I had to establish in my PhD project is the epigenetic profiling to investigate the epigenetic changes occurring during development of pluripotent stem cells towards hepatocyte-like cells. And epigenetics describes alteration of gene function that do not rely on changes of DNA sequence, but modifications of DNA bases or histones. So what you can see here in this figure are the 2 main epigenetic modifications, which is the DNA methylation and the histone modification. And we are interested in histone modification, which can be modified by acetylation or methylation or ubiquitination. There are several ways of post-translational modifications of the histones. And these modifications lead to an alteration of gene function. So they can either activate or repress the respective gene. These epigenetic modifications are misregulated in certain diseases like autoimmune diseases or cancer. And of course, these epigenetic modifications are involved in developmental processes, and that is what we are interested in. There are, as I explained before, there are several epigenetic modifications. One of them are the post-translational modification of histones, as well as transcription factors. These are not epigenetic modifications, but they are also very commonly used in the ChIP assay, which I will explain you later on. And you also have epigenetic regulators. So these are enzymes that either read, write, or erase epigenetic modifications, and these are very important in epigenetic mechanisms as well. So all these factors, so epigenetic regulators or histone modifications or transcription factors, can be detected by a ChIP assay via antibodies raised against the factor of interest. And this ChIP assay is used in my project to investigate the epigenetic changes during the differentiation process, and I am focused on the histone modifications. So you can say the histone code. So how is the ChIP workflow working? I will introduce you to the steps of common ChIP workflows. The first step is the sample preparation. So you have your cultured cells or your tissue of interest, and the first thing you will do is to cross-link the DNA to the protein, so to the associated proteins by commonly formaldehyde. Then you have to lyse the cellular membrane to release the nuclei, and afterwards you have to shear your chromatin to an appropriate size matching your assay of interest. What you have to do then is to perform an immunoprecipitation. So there you mix your sheared chromatin with an antibody specific to the target of interest. So the antibody will bind to the protein, which is cross-linked to the respective DNA sequence, and you can enrich these DNA sequences bound to your protein of interest during this immunoprecipitation step. So after this immunoprecipitation, you have to reverse the cross-links between the DNA sequence and the protein to release the DNA sequence, and afterwards you have to purify your DNA sequence that will be further analyzed in your endpoint of choice, which is commonly qPCR or next generation sequencing. So during sample processing in my project, I faced a serial issue which was caused by the primary human hepatocytes. These primary human hepatocytes are required as a positive control for my hepatocyte-like cells. So these resemble the gold standard. And I wanted to compare my hepatocyte-like cells to these primary human hepatocytes to see how the epigenetic environment is in the in vivo counterpart and how it is in my in vitro generated cells. And as I tried to process these primary human hepatocytes with the common ChIP workflow that I previously used to ChIP my human-induced pluripotent stem cells, this was not working. So my common ChIP workflow was working well in my stem cells, but I'm facing a serial issue in the primary human hepatocytes. And in this figure, you will see the main issue, which is an inefficient shearing when processing the primary human hepatocytes with the common ChIP workflow. And you see here in blue my target size range of DNA fragments. And here is this red line. These are the DNA fragments that I achieved with my common ChIP workflow applied to the primary human hepatocytes. And what you can see is that the gap of the size ranges are almost completely overlapping with my target DNA size range. So that is not acceptable. And what are the main issues occurring with processing primary human hepatocytes is an incomplete cell lysis. So there are still a huge portion of cells that are resistant to chemical lysis. I observed massive cytosolic contaminations in isolated nuclei, so which was visible through a brown-colored nuclei palette. So we now know these brown cytosolic contaminations are, at least in part, lipid droplets that are co-sedimenting with the nuclei when centrifuging, and an inefficient shearing that you see here in this figure. And this inefficient shearing is mainly caused by these cytosolic contaminations that are co-sedimenting with the isolated nuclei, thereby disturbing the shearing process. So my common ChIP assay is not reproducible in the primary human hepatocytes, and I cannot compare my primary human hepatocytes with my hepatocyte-like cells because this ChIP workflow is completely failing. And so we had to search for a solution for this issue, and I focused on the cell lysis and the nuclei prep step of the ChIP workflow. And let's first have a closer look at traditional cell lysis and nuclei isolation methods. Traditional cell lysis is carried out by a chemical and/or mechanical methods, which is working well in many samples, including the human-induced pluripotent stem cells that I'm culturing in my lab, but it's completely failing in my primary human hepatocytes. And I guess that there are other cell types out there that are equally behaving like my primary human hepatocytes. So what you can see here in this figure are primary hepatocytes that are untreated, and in the right image you see the same primary hepatocytes that are treated by chemical lysis buffers and Downs homogenization. And you see that almost all of these cells are still intact, and this traditional cell lysis is largely ineffective in this cell type. Traditional nuclei purification is carried out via centrifugation of your lysate at commonly 500 to 2000 times of G. And as I told you before, the issue is that there are cytosolic contaminations, largely lipid droplets, that are sedimenting at the same G-force like my nuclei. So it is not possible for me to separate these contaminations from my nuclei with traditional nuclei purification. So the main issues are the cytosolic components in the nuclei pellet. And I have observed that this is strongly cell type dependent. So in my hands, the primary human hepatocytes was the only cell type that are showing this issue. And these contaminations are impacting on the viscosity of the lysis buffer. And this will interfere with the shearing and IP efficiency. So in the next slide, I will guide you through my optimization process. And I've started with applying a method called NEXSON to achieve a more effective lysis of the cellular membrane. So NEXSON stands for nuclei extraction via sonication. And in this process, the nuclei isolation is not achieved by chemical lysis buffers or mechanical methods, but it's carried out via sonication. So acoustic energy is used to release the nuclei. The main advantages of this method are that you can standardize your sample preparation, because you can lyse virtually any cell type with this method. You just have to find your right parameters for the sonicator. The second one is the high efficiency of the nuclei isolation, which allows to scaling down your input material. You also have a better comparison across different tissue or cell types. And last of all, it is highly reproducible. And the efficient isolation of the nuclei also allows for high throughput applications. But applying NEXSON alone to my cells not solve my problem, because these cytosolic contaminations are still in the lysate. My lysis was more effective, but I didn't get rid of these cytosolic contaminations when applying NEXSON only to my samples. So I had to include another step into the ChIP workflow, which was the purification of nuclei through a sucrose gradient. So after lysis of the cellular membrane by NEXSON, I overlaid my lysate on a 2 molar sucrose buffer, and through centrifugation with 90,000 times of G, only the nuclei have the correct size and density to move through the sucrose buffer, and your cytosolic contaminants remain on the over the sucrose buffer. So with this method, you can efficiently separate your nuclei from this disturbing cytosolic contaminant. Now I will guide you step by step through my optimized ChIP workflow, including NEXSON technology and the nuclei purification through this sucrose buffer. So you start by fixing your cells or tissue or whatever sample type you have. What you are doing then is to isolate your nuclei. You're doing that by lysis of the cellular membrane via NEXSON and purification of the lysate through the sucrose gradient. So after this step, you have very, very pure nuclei that are containing almost no cytosolic contamination. What you're doing next, you will apply acoustic energy to shear your DNA to an appropriate size range. And these sonication processes that I'm performing with the Covaris adaptive focused acoustic technology that Roman described to you before. And after this shearing step, I have DNA sequences that are cross-linked to epigenetic modifications like transcription factors or histone modifications. And then you have to carry out the immunoprecipitation with the antibodies raised against your protein of interest. And this immunoprecipitation step I carry out with the solid state technology offered by ChromaTrap. and why I've chosen this technology and what it is about, I will explain you later on. So after the immunoprecipitation, you have an enrichment of DNA sequences bound to your protein of interest. And after reversing the crosslinks and purification of the DNA, you can analyze it either via qPCR or next generation sequencing. So I will now show you some results that I've obtained using my optimized ChIP workflow in two different sample types. First, human-induced pluripotent stem cells and primary human hepatocytes. I have processed both cell types with the same ChIP workflow that I've described to you in the slide before. And what you can see here is that for the human-induced pluripotent stem cells, I require only 30 seconds of AFA to lyse the cellular membrane. Whereas in the primary human hepatocytes, I have to use about 5 minutes, which is quite a long time. And this underlines that these primary human hepatocytes are really, really hard to lyse. But with the optimized workflow, it works efficiently, as you will see later on. So I have applied AFA for 12 minutes afterwards, or 20 minutes here in primary human hepatocytes to shear chromatin to an appropriate size. And here you see electropherograms derived from the bioanalyzer that show you the size range of DNA fragments derived with this method. And again, in blue, you see my target DNA range, ranging from about 150 to 800 base pair. And you see that in human-induced pluripotent stem cells, as well as in primary human hepatocytes, almost all of the obtained DNA fragments overlap with my target size range. On this slide, I will show you how reproducible you can do a ChIP assay in primary human hepatocytes with my optimized ChIP workflow. And I will show you that by showing you a ChIP against the H3K4 trimethyl mark, which is a very common histone modification that is associated with actively transcribed genes. And you see here the enrichment at the GAPDH locus. And GAPDH is a housekeeping gene, so you should find a strong enrichment of the H3K4 trimethyl mark at this locus in every cell type. On the y-axis, you can see the fold enrichment above IgG. So my IgG control is an immunoprecipitation with an unspecific antibody. So this will tell you about the degree of background of your assay. And you see in blue primary human hepatocytes that are processed with the conventional ChIP workflow without my modification. And you see that the standard deviation is, I would say, horrible. And I would say that everybody agrees that this is unacceptable. And in orange, you see the same primary human hepatocytes that are processed with my optimized ChIP workflow. And you see that the deviation is very, very low. And this underlines that this optimized ChIP workflow results in highly reproducible ChIP data. So what you can see on this slide is again an immunoprecipitation against the H3K4 trimethyl mark, but this time in human-induced pluripotent stem cells that are also processed with the optimized ChIP workflow. And what we have done is we examined 2 active and 2 inactive loci in this human-induced pluripotent stem cells. And In blue, you see the enrichment for the H3K4 trimethyl, and in yellow, the IgG control. And on the y-axis, you have the percent input. So the input is the same amount of chromatin that I've used for the immunoprecipitation, but without an antibody. So this serves to normalize your chip data. So the active genes are GAPDH again as a housekeeping gene. So we should find a strong enrichment for H3K4ME3 here. And the second one is MYC. MYC is one of the factors that are used to generate human-induced pluripotent stem cells, and should therefore actively expressed. The inactive genes are CYP3A4 and IGX1A. And CYP3A4 is involved in the metabolism of xenobiotics and is almost exclusively expressed in the liver. So it should be there should be no enrichment in human-induced pluripotent stem cells. And the third one is the IGX1A, which is a universal negative control region, and this is located in the so-called promoter desert that is characterized by the absence of any regulatory factors, including histone modification. And what you can see is that you have a strong enrichment for the H3K4 trimethyl for the two active genes, and you have almost no enrichment in the inactive genes. So, and what you can also see is that you have a really good signal-to-noise ratio that can be seen by comparing these blue bars with the yellow IgG bars. So, as I told you before, we decided to pair the Covaris ChIP sample preparation with the solid-state IP technology provided by ChromaTrap to enhance the reproducibility of the chip assay by focused-ultrasonication and through less manual handling during the immunoprecipitation step. So, you see here the Covaris ChIP technology, and these Covaris ChIP kits are working well in human-induced pluripotent stem cells, and I guess in many other samples. But I had to incorporate the two modifications that I've explained to you before into this process. And this optimized sample preparation I have combined with the solid-state technology provided by ChromaTrap. And together, these two provide a very reproducible and sensitive chip assay. And this assay is compatible with high-throughput applications because the Covaris ChIP and the chromatin shearing with AFA, as well as the IP step with ChromaTrap, are compatible with the 96 well format. So our last question was, how could we pair these two kits that they are working together as efficient as possible? And the main question that we had was, how could we find a balance between the shearing buffer in which our chromatin is dissolved after the chromatin shearing with the AFA device and the immunoprecipitation buffer supplied by the ChromaTrap kit, which is required for an efficient immunoprecipitation. So the ChromaTrap protocol recommends at least a 1:10 dilution of the sheared chromatin dissolved in the Covaris shearing buffer with the ChromaTrap column conditioning buffer, which is the immunoprecipitation buffer. So this is thereby limiting the amount of chromatin that can be used in the immunoprecipitation process. Especially for scarce sample types that require the usage of more diluted chromatin, it may be beneficial to be able to use more chromatin in the immunoprecipitation. So what we tested then are 3 dilutions of the Covaris shearing buffer with the chromatrap immunoprecipitation buffer. The first one is the recommended 1:10 dilution. The second one is the 1:1 dilution of the Covaris shearing buffer with the chromatrap immunoprecipitation buffer. And the third one is no dilution of the Covaris shearing buffer with the chromatrap immunoprecipitation buffer. And what you can see here in this figure is again an IP against the H3K4 trimethyl mark in the human-induced pluripotent stem cells. And here you see the different conditions, which are A, 1:10, B, 1:1, and C, no dilution. And again, you will see the two positive loci and the two negative loci. And you can see that in the 1:10 and the 1:1 and the 1:1 dilution gives a really good enrichment over the IgG control, but you have no enrichment in the no dilution sample. And this is true for both positive loci. And for all conditions, you have no enrichment in the negative loci, of course. So what we can say from this experiment is that it is possible to dilute your sheared chromatin in a less amount of chromatrap immunoprecipitation buffer. So you can apply more of your chromatin to the IP step, which is beneficial for scarce sample types and therefore associated more diluted chromatin preparations. But we recommend to use at least a 1:1 dilution of Covaris shearing buffer with the chromatrap immunoprecipitation buffer. So finally, we asked whether our optimized ChIP workflow is compatible with next generation sequencing applications. And again, we used our reliable H3K4 trimethyl mark, and we analyzed the enrichment in the human-induced pluripotent stem cells and the primary human hepatocytes via deep sequencing. And shown in these figures is the coverage of chipped DNA fragments at certain loci in 2 replicates. So here is replicate 1, replicate 2, and we have done these in all samples. And in the last lane, you see the input control, which is the same amount of chromatin without any antibody. So it is the background of your assay. So what we have done is first we examined the GAPDH locus as a positive control for both cell types. And what you see here is a clear enrichment as well in the human-induced pluripotent stem cells and the primary human hepatocytes at the promoter region of these GAPDH locus. Second, we examined enrichment at cell type specific positive loci, which are NANOC for the human-induced pluripotent stem cells and ZerPin-A1 for primary human hepatocytes. And what you can see here is a clear enrichment at the cell type specific loci in human-induced pluripotent stem cells as well as primary human hepatocytes. And here at the bottom, you see the same loci, vice versa, to control if our Chipseq enrichment are specific, because ZerPin-A1 should be not associated with the H3K4 trimethyl mark in human-induced pluripotent stem cells, as well as NANOC should not be associated with active transcription in the primary human hepatocytes. And what you can see here is no enrichment in all replicates in the human-induced pluripotent stem cells as well as the primary human hepatocytes. So this demonstrates that our workflow is suitable for NGS applications and generates highly specific and reproducible data. So I will briefly summarize what you've learned in today's webinar. So pairing the Covaris ChIP workflow and the chromatrap IP technology provides a fast, easy, and scalable workflow. Enhancing the nuclei isolation with the AFA, ultrasonication technology, and the purification of the lysate through a Sucrose Ultracentrifugation process provides a universal workflow for ChIP. So by the AFA lysis of the cellular membrane and the nuclei purification by Sucrose, you remove cytosolic contaminants that can be present in certain cell types. And with these optimizations, the shearing as well as the IP work reproducible from virtually any cell type. And this allows for a standardized ChIP. And challenging primary cell types, like the primary human hepatocytes that I've presented you today, or upon drug treatments, are among different cell types and, of course, different labs. So this is the end of my talk, and thank you very much for your attention. And now we are open for your questions.
We received quite some interesting questions. We have only 10 minutes, so hopefully we will be able to cover it all. If not, feel free to reach out. We will follow the answers to the audience by emails anyway. So the first question is for Tobias. The question is, what are those cytosolic contaminants, and why do they impact the reproducibility on shearing and IP?
Indicate the presence of mitochondria, of course, or other complexes that containing any hem, so such as the cytochrome P450 complex in the liver, and primary human hepatocytes are also quite lipid-rich, so the lipids likely contribute to the contaminations. In general, I think that these contaminations are strongly cell type specific, as I've seen it with the induced pluripotent stem cells and the primary human hepatocytes, and that this contamination differs significantly between different sample types.
Good, thank you. I have a second question for you, Tobias. How sensitive is the AFA chromatin shearing to a variable number of cells?
Yeah, so what I observed, I have to mention that I'm using the M220 ultrasonication device. So I observed that the shearing is quite insensitive to cell numbers, so ranging from about 5 million to 20 million. But what I recognized in my case is that when I'm applying more than 20 million cells, the M220 is some way limited. So I try to not use more than 20 million cells. But in the range of about, I would say, 3 to 5 million to 20 million, the shearing is very reproducible, I have to say, and I'm very it's working well in my hands. Yeah.
Okay, thank you. The next question is probably for Angela. Could you please share technical details regarding sonication? I guess it's for the parameters.
So basically, for all the technical details that you are asking, we have an application note about the work that we have done with Tobias. So you can find it on our webpage in the technical details, and also you can send me an email to discuss. And if you want to adjust this for your cell type of interest, then just please reach out to me. Usually what we have been seeing that after the sucrose gradient, the normal shearing time course that we perform in a true chip kit is good enough. But if you have more complex things, I have seen in the line that somebody was, for example, asking for embryonic bodies or something, please reach out to me and we will find a condition.
Okay, thank you. And we will have also some material posted on the webinar for everything that you want to download. Thank you. The next question is actually for you, I guess. From how much starting material do you think your workflow will be working reliably?
So Tobias has been doing it with around 10 million cells always reproducibly. And actually, we have been discussing this. And scaling down to around 1 million is fine. And after that, I think we would have, because 1 million to 10 million, I think we are good. After that, we would have to try with the scientist who wants to optimize it according to their cell type and amount. I think if you have really little input, you might have to adjust the molarity of your sucrose accordingly. And also, since the pellet is quite sticky, we would have to see with how little input material this is working. And one thing is very important: if you do the sucrose stuff, you always have to use a swinging bucket rotor, because otherwise you get a smear on your tube and you are unnecessarily losing material. But 1 million is fine. Below that, we would have to adjust accordingly.
Okay, thank you. The next question is for Tobias. What are your thoughts on the sample prep workflow? Any step you think require further improvement?
So I think in general, common chip workflows without my modifications are working well for various cell lines or other, I would say, uncomplicated sample types. So the issues begin when trying primary samples, especially ones that are containing high amounts of proteins or something like that, like the primary human hepatocytes. And for these cell types, I think the workflow with the NEXSON lysis and the ultracentrifugation through the Sucrose buffer is a good solution to obtain a very pure nuclei isolate. And I think that with these optimizations, you should be able to process virtually any cell type. Yes.
Okay. The next question is probably for you, but maybe Angela can comment as well. What is the best histone modification marker to consider in a Chipseq experiment for transcription factor to have this as a positive control and show the positive regulation of promoter or transcription?
So from my side, what I would say is that, first of all, here it would be very essential to know what are you looking at. So for a transcription factor, which we consider to go along with active transcription and activate transcription, I think it could be good to look also into K4ME3 to see that you're indeed looking at active promoter regions. And for the gene of interest, you might be looking into K36ME3 also. And if you are more looking into like epigenetic regulators, which could also come along with inactive marks, so you always have to know what is your readout. So if it's more things that are associated also with creating an inactive chromatin surrounding, you might also be looking into K27ME3 or K9ME3. So it really depends what you are looking at.
Okay, thank you. Question for Tobias. How much more time do the additional step add to the normal chip workflow?
So I would say it depends on, I guess that with common lysis methods like chemical lysis or with Downs homogenizers, you have to lyse your samples a couple of minutes. And primary human hepatocytes, I had to apply more than 30 minutes of lysis buffer or even more. And so in the case of primary human hepatocytes, I would say you save time with this workflow because you need about only 5 minutes for the lysis of the cellular membrane. And you need this 1 hour through the Sucrose buffer, but you are saving time at other points of the workflow. So I would say for complicated samples that need to be processed with these optimizations, you would save time comparing to common chip workflows, and you're getting even more reproducible data.
Okay, thank you. Another question for you. Do you have a ratio of cells per ml of buffer that can be efficiently sonicated?
So I commonly use about 10 to 20 million cells per milliliter of lysis buffer. I've tried it with about 5 million, I guess, and I would say you can scale it down even a little bit more. But 10 million cells per milliliter of lysis buffer and shearing buffer is working really well in my hands.
And
okay,
actually, maybe just as an add-on, from 1 to 3 million cells, I would suggest to not do it in 1 milliliter tubes, but to do it in 130 microliter tubes. And from 3 million to 20 to 30 million cells, depending on the device you have, you can do it in the 1 ml tubes.
Okay. I'm going to combine the next two questions. Can we use a speed vac to concentrate the sheared chromatin samples if protein concentration is low? And do you determine the number of nuclei after the nuclei preparation? And were you able to increase the number of nuclei with your optimized method?
So I think using a vacuum for concentrating your sheared chromatin is not a good idea, because you're raising the concentration of your SDS by concentrating your buffer, and that will interfere with the subsequent IP steps. So that would not be a good idea. In that case, I would go the way that Angela suggests, to use smaller tubes and less amounts of lysis buffer to get a more concentrated chromatin. And I've not counted the amount of nuclei in that I've isolated, but I would say that I'm not losing very much of nuclei and therefore DNA by purifying my nuclei through this sucrose gradient. I hope this is a sufficient answer.
Okay, thank you. We have a lot of questions asking about the time it takes to do several steps. So maybe can you remind how much time it takes to do the entire workflow?
From starting with the cell fixation to the IP step and the analysis via qPCR, it takes about 2 working days. 2 working days is realistic.
Okay. And maybe the last question, and again, we will be reaching out to the people who had more questions. Can I skip the sucrose step if I'm using ESC, embryonic stem cells?
Yes, you can skip the step. With my human-induced pluripotent stem cells, the common chip workflows without these sucrose buffer and this NEXSON lysis is working really well. So you don't need this with this cell type. I guess the stem cells are quite comparable to other cell lines that are comparably easy to lyse and shear. I would say you only need the steps when at very certain primary cell types, like primary human hepatocytes, and I guess there would be other cell types out there that are making these problems.
Okay, great. Thank you, Angela, and thank you, Tobias. We have a lot more questions, but we will reach out to bring the answers.
Thanks so much for joining us today, and I'm sorry that we couldn't get to all those questions. Like Roman says, they'll be reaching out later. I really want to thank Tobias for sharing his workflow and his expertise with us today, and also thank Covaris for sponsoring today's event. Thanks for joining us again, and we look forward to seeing you at our next webinar.